Hello, everyone. Welcome to today's live broadcast, What are the Current Advances in Flow Cytometry, presented by J. Paul Robinson, PhD. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. I now present today's speaker, J. Paul Robinson, PhD, the SVM Professor of Cytomics in the College of Veterinary Medicine and a Professor of Biomedical Engineering in the Weldon School of Biomedical Engineering at Purdue University. I will now turn the presentation over to Dr. Robinson.
Well, thank you, Judy, and welcome to all of you to this discussion on advances in flow cytometry. What I want to do before I get into the details of the webinar is give a small background on flow cytometry, and I realize that a lot of you will be probably experts in the field, but there's always some people who are not, and I don't want to go past those individuals. So flow cytometry has been around for a long time, over 50 years, and it started out with just one or two parameters, and that's important for today's discussion because it's now moved to a large number of parameters, from 10 to 25 parameters might be a possibility, and I think will increase over the years. Now, most flow cytometry is going to be around the 5 to 15 parameters, and that's the general area that I'm going to talk about today. So flow cytometry, as most of you know, measures single cells. It's the most unique technology for measuring single cells, and the reason is it can evaluate properties of each population, of a mixed population, without physically separating those cells. That's a pretty powerful capacity for a technology that can look at single cells. And while it does that, it creates a strong basis for statistical analysis. That means that we can actually quantitate information in each of those populations that we might be going to measure. Now, the things that you can measure are either extracellular receptors, typically immunophenotyping, or you can measure intracellular components. Look at DNA or other subcellular organelles, or molecules produced in the cell or on the cell. So there's a lot of things that you can do with this technology. Now, no technology is perfect. There are limitations in everything. And I want to just mention 6 limitations because I'll be discussing most of these during the webinar with respect to the Attune. First of all, sensitivity. The system has to have detectors capable of measuring the number of molecules of the marker that you've attached a fluorochrome to. And so sensitivity above background becomes important. Concentration, sample concentration, and flow rate, those can be really complicated issues in flow cytometry, and they will get you one day or the other that your sample is too concentrated or not concentrated enough, and that will cause you some problem, and we've all faced that issue over the years. Sample volume. What happens if you just simply don't have very much volume? What do you do? And that's an interesting question because when we talk about flow cytometry, I can guarantee you that we always say, look, flow cytometry is fantastic because you can look at such small volumes. And this is true, except that if you only have a few microliters of sample, then it's not so easy to actually run it on most flow cytometers. And then the other issue is the number of samples per hour that you might have. We typically have run tubes over the years in flow cytometers, but today we move to plates, and so we need to look at issues of what is the potential flow rate, what's the sample rate, not just the flow rate, how many samples can you run per hour, not just how many samples can you run per hour, but how many samples can you run per hour and get good data from. Then number 5 there is the number of parameters and colors. And I indicated that, a lot of instruments are now coming on market that are in this range of 5 to 15 parameters, which seems to me to be a pretty popular range. And that's because we've really developed some good assays where we can get assays running with a fairly small number of cells and still be able to look at a vast number of parameters. The last thing is a tricky issue, and that is looking at size of particles, small or large particles. And that's a tough one for a lot of flow cytometers. So looking at very, very small particles is something that I'm seeing a lot of flow cytometry doing. And so I anticipate we'll be looking more at that area. So for the next 45 minutes or so, I'm going to discuss my experience with the Attune. And the last several months, I've had the opportunity of playing with the Attune in my lab, and I've spent quite a bit of time with colleagues who have this instrument, and of course my lab staff who've been using it. And I thought that this would be a good opportunity to outline some of the features that I find attractive actually about the Attune instrument. And I'll discuss some of the unique advantages of the technology, because there are, in every instrument there's usually something that is really quite unique on that instrument, and there's a couple of things on the Attune that I'd like to share with you. And I'll try to expand on some of those issues because I have discussed this with a number of people, and I've found that some people don't quite understand exactly how the technology works. So my goal will be to try to outline that in a fairly easy to understand way. Now, I'd like to put this picture up. It's a picture of Howard Shapiro. I think you probably all know him, and you see his book, Practical Flow Cytometry, there, which I highly recommend. If you haven't got this book and you're interested in doing any type of flow cytometry, you really owe it to yourself and your lab to get a copy of this book. And it's a tremendous book. I call it the Bible of flow cytometry. But there's one thing that's quite unique about acoustic flow cytometry, and that is that it's not mentioned in Howard Shapiro's book. And so that brings me to the interesting discussion now about flow cytometry and how the Attune works, because it has this unique feature of having a different mechanism for aligning cells. And I'm going to spend a little bit of time talking about that because it's one of the most important features, I think, of this instrument. So this is the Attune. It's an analyzer, can measure up to 14 colors and 2 scatter parameters. It's an instrument that has the same general features of most instruments. It has a sample delivery component. It has a fluidic system. It has optics and electronics. And uniquely, it has the acoustic technology. So let's discuss the acoustic technology. Now, I'm not sure that too many of you will be familiar with the Journal of the Acoustical Society of America. And probably most of you don't care and will never look at this journal. But I want to show you some of the things that you missed if you haven't looked at this journal. Now, if you were interested in the acoustics of Italian historical opera houses, you would have seen a wonderful paper in a recent journal issue of this journal. And if that doesn't tickle your fancy, how would you like to be able to measure the height of a speaker in a room simply from the acoustics? Now, when you think about that, that's pretty amazing. How high is the speaker standing at a lectern just from understanding the acoustic information? Well, that's the sort of paper that you might find in the Journal of Acoustic Society of America. But it's not the only paper. And it turns out that a few years ago, Greg Kadacek published a paper in this same journal. And that paper was entitled Ultrasonic Particle Concentration in a Line-Driven Cylindrical Tube. Naturally, all of you would have searched out this paper and reviewed it and read it and would have fully understood it. Well, perhaps not. But had you been looking for this sort of technology, then you would have learned about the beginning of acoustic flow cytometry. So let's look a little bit at the paper that Greg published. So here is a picture of his tube that he made, and on the side here you'll see there's what's called a line source. So what he did was he said, well, I understand what acoustic waves can do. What do they do if I were to put an acoustic device on the side of a tube? And then he put this tube underneath a microscope, and what he saw were some pictures. And I'm explaining it to you here by this little animation. You see that it put a speaker on the side of a tube, it builds standing waves. And if there were dust in the tube, then that dust would end up being in the center of the tube. And that was like an aha moment for Greg, because he realized that the dust particles would be retained in the center cross-section of this tube. And what that did was gave him an idea. And these are the pictures that he showed in his paper. So on the left here, you see before the acoustic device here was turned on, and on the right, you see the particles pretty much aligned in the center. And so this was the start of an idea. Now, you don't transform these ideas into technologies without looking at the theory. And just in case any of you would like to double check Greg's equations, I put them up here. I'm sure you'll agree with me that they look great, and we'll just move on. But the bottom line is that he tested the theory of this and then built a prototype. Now, if you go back a few years when he built this first prototype of the acoustic system, you'll see here it looks, well, pretty much like a prototype, something that would probably end up things that I build in my lab. Interestingly, his first thoughts on this, he brought to people at Los Alamos, and he showed them this idea, and some of the people at Los Alamos said, you know, you could do this you could build a flow cytometer with this. And the original idea was maybe you could do this without having sheath fluid. And that was the original concept of building a sheathless flow cytometer. But as you develop technologies, as I will explain to you in a moment, things don't always work out the way that you start. Here is some more material published by Greg, and I want to show you what the effect of this transducer over here is. And on this side, there's a cartoon, and on here is a real image. This is actually originally a video, but I couldn't show the video on this system, so I broke up the video into some small sections. So I'll show you, first of all, there we have as the system starts, and this is before acoustic focusing is turned on. And now what we're going to do is turn on the acoustic focusing. So turn it on just a little bit, and you see the cells starting to move to the center. We turn it on a little bit more, and the cells are further in the center, and you can see over here on this side that they're really starting to align quite nicely. And now we've got the acoustic system on full bore. And look at this alignment. They're just perfectly aligned. And over here, the real image, you see that the cells are in the center of the core. And here, of course, is the acoustic waves as they go through the system. So from these early experiments, Greg was able to show, wow, this really will work on a flow cytometer. And there are some advantages to this. And that's really the key point of this webinar, is to explain some of the key advantages of acoustic flow cytometry. Now, you're all, I think, familiar with hydrodynamic focusing. This is what we do in almost every machine. We have a sheath fluid that goes through here. We have a tube here, a sample delivery tube, and the cells in here are sort of all over the place. And what we do is we focus them down through this, what Howard Shapiro calls this neck down region here. And they focus down, and we have pretty much single cells. But if you increase the pressure on this to go faster with traditional hydrodynamic focusing, you don't end up with a good situation. And we've all seen that in our systems, that you lose some of the integrity or the quality of the data. So what's the difference in an acoustic system? And I want to make a pretty important point at this stage. The current version of the acoustic flow cytometer, in fact the commercial instrument that was built, wasn't exactly the same as the original concept that Greg developed. He developed a concept that was basically sheathless. Sounded like a good idea at the time, but practically it is not quite as good as a sheath-based system. And what Greg discovered is when you put the two systems together, that is, you have both the hydrodynamic focusing and the acoustic focusing, you get the best of both worlds. Now, you probably can get away without hydrodynamic focusing, which is true because you do this in many of the commercial instruments that are out there which don't have this technology. And that's fine when you're going reasonably slow, but the powerful advantage of the combination of acoustic focusing and hydrodynamic focusing is that if you decide to move this system at high speed, then this is where the real advantage is, and I'm going to spend some time talking about that. So let me reiterate a couple of things by showing you some really old slides. These are slides that have been in my class deck for over 20 years. And I try to explain with this slide the concept of hydrodynamic focusing, of the core here and cells coming down the core, and the fact that we have a laser coming through here somewhere, and at about 90 degrees, we measure the fluorescent signals. And this is all very well, but in fact, in most analyzers, we don't do this. We actually do this. We actually turn the system upside down. We flow upright. And the only time we really flow down is when we're sorting. It's a little bit difficult to sort going up because, well, it would be rather crazy. But for analysis, it's far better to flow up. So just wanted to refocus us on how this concept works, because this helps us see the difference between a traditional instrument which has hydrodynamic focusing and an instrument that has both hydrodynamic focusing and this acoustic module on the side. And so the advantages here are that by the time the cells reach the hydrodynamic focusing area, they are already in line. And this allows you to increase the flow rate significantly without losing integrity of coefficient of variation, which, as we all know, is important. So let me summarize a little bit here for what we've been discussing. Why does this matter? I always like to ask myself that question. Why does it matter? Why do I care? So the instrument gives best of both worlds, because we know that hydrodynamic focusing works well in most instruments that are out there. But when you add acoustic focusing, you get a new dimension. And you'll see that in a little while when I show you some different data. And the highly stable flow at high speeds is quite difficult to achieve on most instruments, but that's definitely one of the advantages that we see in the Attune. So let's move on and discuss some of the other features of the instrument, because there are some things that I quite like about the instrument and that I think makes it a very attractive instrument. Let me move to the optics and electronics. So if we take the cover off, which I suspect you're not supposed to do, this is what you will see. On the left there, and let me block out some of this so that you just focus on some parts of the instrument. There's a lot of stuff here. So we have some preamplifiers. You see a bunch of preamplifiers on this instrument, and the photomultiplier tubes are inside there somewhere. And then we have a bunch of dichroic filters that you can see here, and a stack of bandpass filters. So this is the core optical assembly that you have on the instrument, and you see here there's a fiber optic coming from somewhere or other, and that is addressing the detectors from the detector system. So coming back to that original picture, this is now showing us where the lasers are. And we have the possibility of multiple lasers. The instrument that I have in my lab has 4 lasers, but I understand that you don't have to have 4 lasers. You can have 3, 2, or 1. So the blue laser is generally in that position. The yellow or green laser is there. The red laser is over here. And the violet laser, or at least the laser we call the violet laser, it's a 405, sort of looks a little bit violet, is down in the front of the instrument. And if you look at the path that those lasers follow, it's pretty much like I've outlined here on this graph. And you'll see that the lasers are actually physically separated from one another by a physical distance, so that the cell goes through each each laser successively. And there's some advantages to doing that, which I'll discuss in a moment. But let's, for a second, look at these lasers. They're quite interesting. First thing that I noticed when I pulled the lid off the machine is that the lasers have a slightly different structure to what I was used to. The laser itself is hidden underneath the instrument at the back, but the delivery comes here into this optical assembly here. And you have this round system here that is an optical assembly. So what are the lasers? They're actually an OBUS coherent laser, which has been designed particularly for this instrument. Excuse me. So there are some unique features about the laser. And the first feature is that it's fiber optic delivered into this big blob here. And the question is, what is in that blob? So let me clear all these marks that I've made on here so we can look at this optical device. There's a number of features. The laser comes in here, and then there's a collimating lens that focuses the laser, and then there's this thing called a power lens. And what that does, as I'll explain in a moment, is it spreads the beam out a little. Instead of it being a perfect Gaussian beam, it gives it a little bit of a wider spread. And there's a value in that that's quite unique in the way that the Attune uses its laser to focus. And then there's a telescope and a doublet lens here. I'm actually going to jump this slide and come back to it. This is what the laser signal would look like if you were able to see this on your machine. Actually, the specification of the laser is such that it has to have this format of beam. We call this a tophat, or the Attune uses the term flattop. There's an advantage in this because, as you know, we often broaden the laser out a little bit so that when a cell goes through the laser beam, that it gets an equal amount of intensity. We don't like having the intensity of the laser beam, which of course is the strongest right here, but it falls off exponentially as it goes down here. So there's a huge difference in intensity at this point if the beam is shaped this way. But that's not the same problem if you have this tophat. And it doesn't really matter if the cell comes in at any of these places. It's going to get the same amount of intensity. Now, you might not care about that, but in fact, the quality of your data will change significantly depending on what you're running if you are misaligned a little bit. Now, I'm sure you could still be misaligned on any instrument, but the bottom line is that you have a better chance of getting better data if you have this broad beam. So, for example, in cell cycle analysis, which I'll mention later, that's actually critical, and you need to be very, very careful. And we'll talk about why the Attune, I think, has some really nice advantages there. Now, in talking about the laser and the laser quality and the signal quality, it's something that people have talked about for years and years, and I think is now becoming more common in instrument design, that you get the Levy-Jennings plots for every one of your detectors, and the Attune is no different. It has that feature, which is very, very important quality control. If you want to review whether or not your instrument has been performing, this is the way to do it. And I strongly recommend that you take note of those plots if you have not been doing so. Something that's very easy to see on the Attune, and I strongly recommend it. So why does it matter that we care about the quality of the laser, the quality of the signal, the reliability of the alignment, etc.? Well, it matters a great deal because the goal of most flow cytometry is to get a good quality CV, the best that we can possibly get. And that's what we should strive at. Now, I know it's hard to get great CVs in phenotyping, but that doesn't mean you shouldn't strive to get high quality CVs. Cell cycle, it's critical. So that's something that you should take note of. And the stability of the signal intensity certainly makes it important for experiments performed over days or months. You want to compare results. You certainly want to know that the data are compatible from one day to another, or comparable, which is probably a more accurate way of saying it. So if you want to be sure that the data are the best, you should strive to calibrate your system and make sure that you keep all of these things tuned appropriately. And that's, of course, a scientific goal that I think everybody has. Well, there's a couple of other things that I think are definitely worth discussing on the Attune, and one of them is the fluidic system. There's a interesting, going back here to another slide that I have had for many, many years. The only modification I've made in this slide in 20 years is I added the peristaltic section here, because that's been something that I've noted in the last several years in terms of delivery of samples. But traditionally there were two types of systems. There was a positive pressure system and there was a positive displacement syringe system. These have been around for decades. And you know that in the positive pressure system, the key to stable flow here is having an appropriate differential pressure between the sample pressure here and the pressure on the sheath flow in here. And we know that to get nice sample flow here, stable sample flow, non-turbulent flow, we need to regulate the pressure here very carefully, and we get this very small differential, and that's how we basically change the rate of flow of cells. It's very different in a positive displacement system. In a syringe system, which is what the Attune is, it's quite different. First thing that we notice is that there's a syringe here, and there's a sample loop of some sort, and then there's your sample. So the goal here is to pull the sample out of your sample tube into some kind of sample loop, and then you change a valve, and then you deliver that at a constant flow rate, and your sample's going through the system. Now, the advantages of the syringe system as opposed to a positive pressure system is that syringe systems are innately quantitative. You can measure the absolute flow rate or the absolute number of cells because you know exactly the volume, you know exactly the length of time that it's run. So that's actually really important. Now, I haven't changed this in many years, but this is really no longer true. The Attune can go much, much faster than this. So that's actually a significant change in the Attune, and something that I'll address again in a moment. So the concept of a syringe system allows you to do absolute counts. So this is just a photograph, again, taken the back off the instrument. I'm not sure that you're supposed to do this, but unfortunately, when an instrument comes in my lab, I tend to pull it to pieces to see how it works. And this gives me a very good understanding of how the instrument actually performs. You can see the quality of the construction and how the whole system is designed, how easy it is to repair. And this is the photograph I took of the back of the system. And over here is the syringe. So this is a syringe, and you put the sample into your sample tube, and that's somewhere over there. It's sucked into a sample volume, and you might wonder why there's all these parts of this system. But this valve is a fairly complicated valve. And that valve is allows you to pull up sample into a sample loop. It has to have ways of then delivering sample, of cleaning, etc. But this is on the side of the instrument. This is the part that actually delivers the sample to the instrument. And the fluidics environment is over here. Now, something very interesting here about this system. And I mentioned earlier that the original idea of Greg's was to build and design a system that was sheathless. Well, it didn't turn out quite that way. It isn't sheathless, it's actually less sheath. So that shows that this is the sheath tank, and it's really very small. It's tiny, and this suggests that the instrument is capable of going for extended periods of time with a very small amount of sheath. So that's a nice feature about the system, and that whole sheath tank is right here on the front of the instrument. Now, while I'm looking at this slide, I haven't discussed the automation yet, but if you had a plate reader on the side of the instrument, it would be sitting here. I draw your attention to this chamber here. If you open that up, you'll see that there's a sheath tank here. So this also is very, very small. These are about 2 inches, maybe 60 millimeters, 50, 60 millimeters in size, 2 or 3 inches, very small. And so from a sheath system, the instrument takes very small. And just another reminder of the fact that because you have this syringe, you have volumetric delivery and absolute count. So again, let me ask the same question. Why does it matter? Why do I care? Well, absolute counts will actually save a lot of money. If you don't have an instrument that does absolute counts, then you'll have to buy expensive beads and calibrate those beads. And so there's an advantage, I think, in doing absolute counts and using a syringe system. The other thing is it's a tremendously stable flow, and you have highly controlled sample delivery. And the other advantage, I think, is that you can go from slow to extremely fast. So let's look at the actual sample delivery. And this has got some interesting features that I noticed on this instrument too, that I found that were quite attractive. So this is the sample tube, and the system is pretty much the same as other instruments in that you can stick a tube on here. It's a spring-loaded system. It pops up, and the sample collection tube is in the center of the tube and does what most systems do. It just sucks the sample up into a loop, and you're good to go. I show you this picture for another reason, and that's because you see a second tube coming here, and this, of course, is where your plates are. What's interesting about this system that I know in my lab we run a lot of plates on the plate reader, and one of the things the staff like to do is instead of using a well on the plate to set up the instrument, they like to do it the more traditional way, is take some sample, put it in a tube, put the tube in the machine, and run that tube, set up the voltages, get everything right, and then they're happy that they can then switch over, put their plate in, and go. And the advantage is that hidden underneath here is a valve, and that valve has a link between this and the system. And so by simply pressing a switch, you can switch from here to here, and you can run the system. That's actually a really, really nice feature. It's a well-engineered system. It's a lot of thought gone into the fact that, often the time you'll use a tube to calibrate a system and run a plate on your system. So a few features that I mentioned earlier: reasonably good dimensionality in looking at small particles. It has the capacity to use the 405 nanometer laser for scatter. And of course, because you reduce the wavelength of the laser to 405, you increase the resolution. So that's a nice feature. But I really want to focus on this feature here. You'll see, not bad resolution here, but the real point that I want to make here is that when you have the acoustic system that you can run at really quite high rates, even faster than that, but this is a very high rate of sample flow, and get quite excellent resolution. The other thing is, because, as I noted before, the beams are separated, you can run a 561 laser on this instrument, and because of that, you have separated beam, so you can look at your FITSI and your PE signal with different lasers, and so you can avoid compensation. And that's always a handy feature if you can do that. So I just wanted to point that out. So again, why do all these things that I've just discussed matter? Why do I care? Well, I like the mechanism for being able to switch from tube to plate without doing anything but pressing a button. I think that's actually quite nice. And also it's very easily accessible. It's open, so that putting a tube in there, so the technicians in the lab like that very much. And you can flow moderately fast without loss of resolution. I might actually change this to very fast or extremely fast. Moderately is probably far too conservative a word there, but it definitely is very, very fast. And having onboard QC, etc., will certainly help you. So there's a couple more features that I'll mention in the last few minutes of my presentation. And one of these is automation. I've already shown the fact that you can put microteeter plates in here and run your microteeter plates directly into the system. It can be permanently hooked up, and in my lab it is permanently hooked up, and we switch frequently from running tubes in here to running plates in here. So that's the first feature, but a feature that I haven't yet tested, but I have seen it in operation out in Eugene, Oregon at Thermo's site, is their automation system. So what they've done is they've taken their orbiter system, which can sort of grab a bunch of plates from here and pop them in here and then put them back. And my understanding that this can be done in temperature-controlled environment, in heated, cooled, whatever. And you can, of course, use an incubator, an automated incubator here. So this is a system I haven't tried or tested, but I have seen it in operation. So I understand that this is the next thing. So I'm sure that there are plenty of people out there that may well be interested in that as a feature. And I will point out that, of course, you can't use this small container for sample fluid if you're going to run vast numbers of plates. You've really got to have a bigger system. So I put these two pictures there almost in comparison of size. This is sort of a relative size, and this is quite a large container that sits on the floor, and that's required for the high number of plates if you were going to run the fully automated system. So that is there. Again, haven't tested it, but I've seen it operating. So why does this matter? Well, I think if you're in an environment where you wanted to run a large number of plates, the one thing that's for sure is that automation is a very good thing. Running plates, I've found over the last several years that I've been running a lot of microheater plates on flow cytometers and preparing a lot of plates using automated preparation systems, that there is a reduction in error. So robots do exactly what you tell them to do. They are extremely reliable. They don't get sick. They don't charge overtime. They'll work all night if you want them to, and they repeat things with precision. So if you're doing preparation of microheater plates, and I suppose if you have one plate to prepare, then it's probably not worthwhile. But if you have 3 or 4 plates to prepare for an assay, there's no question in my mind that automation improves the quality of the data because the volume distribution is precise. It facilitates timing. If you have to add timing in your assay, if you're doing phospho assays or something like that, you've got to stop the reaction at a certain point, and it's got to be precise, and you want the same every time. So that has to be done, and robots are very good at doing that. Okay, so let me now briefly mention cell cycle analysis, and I've got just half a dozen more slides in the webinar. So cell cycle analysis is something that we've been doing in flow cytometry for literally 40 years. It was the tool of choice in early flow cytometry days. You know, there was a day when people didn't do much else but cell cycle analysis. And one of the things that you know for sure if you do cell cycle analysis is that you cannot run samples quickly. It is the slowest, probably most boring type of flow cytometry because you've just got to run things slowly. And the reason is you need good CVs. If you don't have good CVs, there's no way you're going to tell the difference between a very small change in nucleic acid because that's what you're measuring. So we know that it takes several minutes per sample. It's very time consuming and therefore it's expensive. It's not expensive for reagents. They're not expensive. It's the time, the technician time and the lab time and the flow cytometry time that will really kill you if you're going to run cell cycle analysis. Well, that model doesn't work with the Attune. In fact, it's completely changed. And I'm really quite amazed at this because of the acoustic focusing, it's a total paradigm shift with the Attune. And my suspicion is that people who aren't doing cell cycle analysis, simply because of the reasons that I've mentioned, will do it if they have this instrument, because it's almost a pleasure to do cell cycle analysis. You can run those samples through at much higher speed and get high quality CVs. And if you do this on a plate, and this is something that I never thought I would see, but if you run an entire plate of cell cycle, well, there's several things that happen. First of all, the analysis is a cinch. One of the things that I've learned over the last several years of running lots of plates is that software is not well defined to look at plates. Why, I don't know, because it's a perfect opportunity. You know where everything is. Plate formats are fixed. You've got 96 well, you have 384 well, etc. So when we looked at cell cycle analysis, one of my staff looked at this and just wrote some software that analyzes cells. Why do you want to look at cell cycle analysis like this? I'll tell you why. This is a very, very simple reason. So let's say you're interested in seeing whether or not there's a change. Whoops, I've got the wrong marker. I discovered that you can actually draw a straight line if you pick the right one. But look at this. You can look to see whether or not there's a change in your G0, G1, or G2M. Now, of course, you're just looking at the cells, but what you're doing here is you're saying to yourself, oh, which ones should I look at in more detail? And you can choose the ones that you want to look at in more detail, or you can ignore some of the ones that nothing much happened. So this is one reason. The other reason is, sure, it's all very well to look down that way, but one of the things that my staff member did when we looked at this was he said, well, just click a button and rotate everything so that they're actually going in this direction. And now you can look at the rows as well, changes in rows as well as columns, because it never occurred to me when I looked at this that, yeah, it's all very well to look down the column, but there are things called rows as well. So that's just one thing. And the Attune is able to do cell cycle analysis in an entire plate. Again, something that I never thought anyone would ever do. Just to show you the impact of speed. And here we have very, slow, normal speed, very fast, what I called really fast, and something that I've termed ridiculously fast. I would never believe that anyone would run cell cycle analysis at this sort of speed. But look at this, absolutely superb CVs at this rate. And can it be done, and can you do really good work? Well, I'm going to show you something from my friend and colleague, Professor Jake Jagerberger. This is really complicated stuff, and Jake showed some of this data at a recent meeting and was very generous in sharing some of his slides with me, which I understand are going to be published very shortly in this book that I've referenced here. But look at the detail and quality of the work that Jake did. And Jake did all of his cell cycle analysis on an Attune and running them on plates. And just absolutely gorgeous work in great detail. This tells me that this is a transformational moment in cytometry, that you can do cell cycle analysis at moderately or at high speed. Certainly the sort of speed that you can do your cell cycle experiments in an entire 96 well plate and run them automatically, and then have a very rich data set to analyze. This, to me, is a transformational thing in cytometry. So why does all this matter? And I'm going to finish up with a summary in my next slide. Why does all this matter? Well, cell cycle is something that I've always sort of hated doing. And not just me, the technicians in the lab don't like doing it at all because it's so slow and everything has to be so perfect. And you've got to be so worried about blockages and keeping the flow rate nice and stable. But actually, that's not the case with the Attune. And you can get excellent CVs at really pumping those cells through the Attune very fast. And I really am amazed at that. And I wouldn't have thought that was the case until I sat down and actually saw the data. So that's why I think it matters. And let me now finish my seminar and summarize some of the things that I've said. Now, I've really tried to, in the last 55 minutes, just give my personal impression of the instrument, how we use the instrument, what I like about it, what I think are important. And so the acoustic focusing feature, something that I heard about and it's been around for several years now, and I really had to sit down and think about and actually have a talk with Greg Katachek just to find out exactly how did this work and why is it so good. And I'm really convinced that it's a really excellent piece of technology. The fact that it also is combined with a sample delivery system that can provide absolute counts, and that you can combine both of those things with hydrodynamic focusing means that you get excellent data. That the laser lines are delivered with tophat that's really finely tuned in a way that alignment is less going to affect your results. And if you do phenotyping, you want to do a lot of samples, you can look at 14 colors with 4 lasers. And this separation of the 4 beams means you have less compensation issues to deal with because you can set your system up in a way that is easier to manage. The ability to switch from tubes to plates, I think it's a really clever engineering feature. And the other thing that I didn't spend any time on this seminar, but I'll mention it briefly now, is that you can run incredibly dilute samples. And I heard a story of that Greg told me that someone had dropped a sample on the floor and there was almost nothing left in the sample. And Greg said to the scientist, oh, look, just put some saline in there and we'll run the sample. And they did, and they got great data out of it. And the reason is you can run that volume through the system in a reasonable time, and by the combination of acoustic and hydrodynamic focusing, you're going to get excellent quality data on very few cells. Finally, you can run cell cycle on this system at a very fast rate and get a good reduction in CV. And I've already mentioned the automation with the robotics, and that may well be of interest to a number of people. So let me finally acknowledge the assistance I got from Jolene Bradford at Thermo for her support for our laboratory, which I gratefully acknowledge, for Greg Katachek and Michael Ward from Thermo, who put up with me for a few days to literally pull the Attune to pieces and let me see the inner workings so that I can understand how it works and give me a better understanding of that instrument. I really appreciate that support. To Kathy Rajab, who's a flow technician in my lab, who's running the Attune and runs our data, and Jenny Sturgis, Valeri Petsurkin, and Bartek River, who support my lab and give me the assistance that I have to produce the data that we publish. And with that, I'm going to hand over to Judy, and I thank you for your attention during this webinar.
Thank you for that presentation, Dr. Robinson. We want to get right to your questions and input, so here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your question due to time constraints, Thermo Fisher Scientific will reach out to you via email. And our first question is, Why can you do cell cycle faster on the Attune NxT? I thought cell cycle was always run at very slow cell analysis rates.
Well, that's a great question. Thank you. I've sort of spent a bit more time on that than I see that we're already at 59 minutes here. But the bottom line is, because you can align with both hydrodynamic focusing and acoustics, you can guarantee that those cells are perfectly in line and that you can go faster. That when you go faster, you don't just broaden the core by allowing the cells to move away. They can't, because they're focused in the center by the acoustic system. And that's the real reason why a cell cycle runs faster, something that I hadn't thought about before.
Can you clarify that the Attune NxT has both acoustic focusing and hydrodynamic focusing, and why are they both needed?
Well, as I said before, the original concept that Greg had was that you really didn't need anything else. You didn't need acoustic hydrodynamic focusing, because you could actually go with a sheath-less system. But as he found out when he designed and built the instrument, it works better. The bottom line is that the combination of the two just works better. And when you build systems and test them and you prototype them, you find out what works better, and that, I suspect, was developed through many prototypes. And the bottom line is that we know that hydrodynamic focusing works well, and if you combine the two, then you get a much better system.
Thank you. Wonderful questions coming in. We have time for just one more. Why did you say that the Attune NxT can look at samples with very low cell counts? It seems to me this is a fundamental problem with most flow cytometers.
Yes, that is true. And you really get worried if you have very few cells in the sample. And we've probably all been in the situation where we've got very few cells, but we want to get a sense of what the measurement is without losing our cells. And the problem is that very difficult. So on the Attune, the reason is that because you can acoustically focus it, each of the cells that you have, you can force those cells to remain in that perfectly alignment thing, and you can have them coming one after the other at long distances, and still, so in other words, you have very dilute sample, but you can run that sample at a high volume rate. So if you ended up with 2 mils of sample and you only had 50 cells, with most flow cytometers, you would be sitting there for a long time. You would just not be able to find those cells. But with the Attune, you can run at a high volume, several hundred microliters per second, and still be sure that those cells are going to come through and focus. And so that's the reason that you can run at a high volume of sample and still focus on those few cells that are there.
I would like to once again thank Dr. Robinson for his presentation. Do you have any final comments?
No, I've enjoyed the opportunity to do the presentation. I'm sure that if I've gone a little over time, I apologize, and I'm sure questions that come in, I'll be more than happy to answer those either publicly or privately. And the links to my website and my YouTube and Facebook are there, so people can find me and track me down.
Well, thank you again to Dr. Robinson. I would also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand until November 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Bye.